In this video, we are going to be going over the answers for the worksheet for Chapter 13. We are first going to be matching some terms and their definitions. When it comes to this, gel electrophoresis is going to be the letter G, which is a technique to separate molecules in terms of charge and size. Number 2 is the letter C. A chimeric plasmid is going to be a biomolecule that is formed from the ligation of foreign DNA and a vector. Remember, a vector is also called a plasmid vector. 3, PCR is the letter E. This is a technique that involves the amplification of the DNA of interest. Remember that this is only a segment. Number 4, ethidium bromide is letter F. That is a molecule used to visualize DNA. Number 5, transformation is letter B. It is the process of intercellular transfer of genetic information, and we utilize it in order to follow the steps from cloning. It's one of the steps that we need to do in order to find the correct construct of our chimeric plasmid. Number 6 is letter A. A restriction endonuclease is an enzyme used to cut double-stranded DNA. Number 7, DD and TP is going to be letter D, which is specifically a biomolecule that lacks a hydroxyl group on carbon 3 in its sugar motif. Let's move on to part B. In part B, as you can see, we're going to have different DNA samples, and we have to give them the order of migration from top to bottom. Understand that these different vectors, as you can see, we're given the size in base pairs. On the left of the figure, we have a 1 kb ladder, and that is just a standard for DNA molecule size. And as you can see, when it comes to this problem, we're going to be lining up from large molecules. And as we move down in this agarose gel, we're going to be moving to small molecules. So, lane 2, we have a vector that is going to be around 2680 base pairs. So if we look at the size, that's going to look, and this is all relative, so it's going to be somewhere here. On lane 3, we have a vector that is around 3140, so it's going to be a little bit higher. On lane 4, we're going to have a vector that is 5371. So that should look like it is around here. Then we're going to have on lane 5 a vector that it is 4952. So it's going to be a little bit lower than the one that we just drew. And lastly, we're going to have a vector that it is about 4505. So it's going to be a little bit lower than the one that we just wrote. Let me just make them a little bit darker. A little bit too much. Lane 3 around 3000. Remember that whenever we're doing gel electrophoresis, our samples are going to look like a band, like a lane. So this is how our samples is going to look if we run them in a gel electrophoresis experiment.